your cells could soon keep an accurate record of their entire biological history, including attacks from microbial invaders or exposure to harmful chemicals. By tweaking DNA, U.S. researchers have been able to store markers of biological events inside the cells themselves and read them at a later date, essentially turning cells into analog recorders. They believe that capturing the memories of these biological events could help to shed light on disease and even unlock the secrets of how cells develop from an embryo to the billions of adult cells which make up the human body. Synthetic biologists at the Massachusetts Institute of Technology made the cellular recorders by altering their genetic material. Previous efforts have shown that it is possible to encode digital information within cells, with their DNA showing whether or not an event took place, such as exposure to a chemical. But the new approach is analog, capturing far more information. According to the MIT Synthetic Biology Group, its approach enables the cell's DNA to capture how long an event lasted for as well its intensity. To enable a deeper understanding of biology, we engineered human cells that are able to report on their own history based on genetically encoded recorders, said Dr. Timothy Liu, a bioengineer at MIT, who led the research. Dr. Liu's team used the powerful gene editing technique CRISPR-Cas to alter the DNA of human cells. This high-precision method, borrowed from bacteria, can be used by scientists to make accurate edits at any point in the DNA they choose. In bacteria, CRISPR helps the microbes keep track of past viral invaders by inserting short snips of virus code into a set region of their own DNA. This acts as a permanent record and helps the bacterium to recognize these invaders in future so they can mount a much faster response. We wanted to adapt the CRISPR system to store information in the human genome, explained Samuel Purley, one of the team's researchers. When this molecular tool is used to edit human DNA, researchers produce a piece of guide RNA, which sticks to the target site of the DNA and tells the cutting enzyme where to snip. But in order to record analog memories inside cells, the MIT team tweaked this tried-and-tested toolkit, by making guide strands to match the DNA which encodes them, creating self-targeting guide RNA. This means that when the guide strand leads the Cas9 cutting enzyme to the target site, it cuts the section which encodes for that same guide strand, causing a mutation in the DNA. The mutated DNA produces a new mutated RNA guide strand, which causes Cas9 to snipping that section of DNA, resulting in another mutation, and so on. By linking the activity of Cas9 to specific circumstances, so the cutting enzyme only kicks in under certain conditions, such as exposure to chemicals, or inflammation, the team could create a permanent memory of the event in the cell's DNA. In effect, it means that as long as the enzyme is active, it will continue to cut the DNA and produce new mutations, which remain in the DNA as a permanent record of the event. And the more mutations that accumulate, the longer the event lasted. In experiments, the MIT team was able to generate a DNA record of inflammation in mice. They were able to engineer a circuit in which Cas9 only becomes active when a target molecule called TNF-alpha is present. As TNF-alpha is present during inflammation, it could be used as a record of how long the inflammation lasted, with a greater number of accumulated mutations in the DNA being stored as a permanent memory of the event. This is the rich analog behavior that we are looking for, where, as you increase the amount or duration of TNF-alpha, you get increases in the amount of mutations, said Dr. Purley. According to the team, the approach could initially be used in humans, programmed to record a number of events such as inflammation or the presence of other molecules, such as carcinogens. With this technology you could have different memory registers that are recording exposures to different signals, and you could see that each of those signals was received by the cell for this duration of time or at that intensity, added Dr. Purley. That way you could get closer to understanding what's happening in development.